Hello, I'm Iraj Ali, CEO of Achilles, and I'm delighted to have this opportunity to tell you a little bit more about what we've been up to. Achilles is a clinical stage company developing precision T-cell therapies to address multiple solid tumor cancers. We have two studies open right now, one in non-small cell lung cancer and one in melanoma, with a plan to bring a third program into the clinic in 2022. The studies are both open and running, and we plan to bring 10 patients of interim data out in Q4 of this year, and we'll be at SITC presenting some of that data. We'll also open a checkpoint combination arm in our melanoma study and move to our higher dose cohort across both studies in Q4 of this year. The design of the process, manufacturing process, is to be closed and automated, and this we believe will enable the delivery of significant number of doses to deliver these indications at commercial scale. The science behind what we're doing is based on fundamental research in the field of tumor evolution and immune regulation, and these ultimately fuse together to unlock a new class of cancer antigen, the clonal neoantigen, that we believe can unlock transformative potential for our patients. We have about a team of 200 employees. We're headquartered in London and fully financed through to the delivery of the first-in-man studies and the build-out of the manufacturing infrastructure and bringing the additional programs into the clinic. So on slide 3 here, you see the overview of the process and the concept. The patient, Mrs. Jones, comes into our process, and we would procure tumor and blood, and from which we'll do two things. The first is extract DNA to perform a genomic analysis, and that's a comparison of the tumor DNA with the healthy DNA. And this identifies these mutations, and we're looking for a special class of mutations that are present on all the tumor cells and completely absent from the healthy tissue. And those are called clonal neoantigens, and we'll talk a little more about those. But let's say if Mrs. Jones would typically, in let's say the lung cancer study, have about 100 clonal neoantigens, we'll manufacture peptides for all of those 100 clonal neoantigens. So we don't do any in silico selection of those antigens, we'll manufacture all of them. From the tumor, we'll harvest T cells in a manner analogous to what you'd see in a standard TIL therapy. From the blood, we'll do something unique in this bottom arc, and we'll manufacture the patient's own dendritic cells. And then we can combine these components, the peptide with the antigenic information unique to the patient, Mrs. Jones, the dendritic cells, also unique to Mrs. Jones, and the T cells, which have been extracted from her tumor. And in the T cells, they would be polyclonal. You could have hundreds or even thousands of different clones. With the dendritic cell and the peptides, we will activate just a subset of those that recognize the clonal neoantigens, and they'll be activated through their TCRs. So you achieve selection and activation in a single step, and that's what drives the manufacture of the product, the clonal neoantigen reactive T cell, or CNET. So the concept behind the target is really fundamental in basis. We've learned over the last few years that tumors are clonal in origin. They start from a single clone that then develops intense genetic heterogeneity over time. That may be 10 years of smoking or 20 years of sunlight. And so what starts off as something that is heterogeneous, so this cluster of red cells, becomes a plethora of different cells that are very difficult to treat clinically. With our technology, we're able to elucidate the phylogenetic tree of the cancer and identify those original red mutations at the trunk of the phylogenetic tree and the clonal neoantigens present in all tumor cells. And that's in all the primary and all the metastatic tissue, and those are what we target and go after. We've industrialized that capability in our platform, Peleus. That has been built with exclusive access to a study called TraceRx, and that's a significant study of over 780 patients. It's about a $20 million investment from CRUK, and in non-small cell lung cancer. And that data has been collected over 5 years, generated multiple significant publications. It has over 3,000 tumor samples. It's one of the largest data sets of its kind anywhere in the world. With that, we've built the unique capability to identify these clonal neoantigens on a per patient basis. So looking across the field of cell therapies, there are many good companies out there going after a number of different approaches. And one way to segment them is to look at the antigens that they're targeting. So on the far left, we have the tumor-associated antigens. And these we've known about for a long time. And they are present on some healthy tissue, and they're not present on all cancer cells. But there are a number of good modalities that you can use to go after them, and then they're represented by some very good companies. What we're doing is slightly different. We're going after a different class of antigen, a personalized antigen, or class of antigens called the clonal neoantigens. And we have a unique capability to get there with our exclusive access to TracerX. And using a TIL-based process, we believe we can deliver a product with higher specificity and higher potency than a standard TIL therapy. So how does our platform do this? We deliver both precision and potency, and that's through our VELOS manufacturing process. We're prospectively targeting neoantigens or using those peptides. So we already know the targets that we're going after in each patient. That allows us to quantify the component of our active ingredient in each product, our CNET. So we may have a product maybe 50% pure for CNET, and then we can track those CNET cells in the patient post-dosing. And we'll show you some data that speaks to that. And this, of course, is going to help us unpack for what has been quite a while in the TIL field, but actually to fully and more detailed understanding of the mechanism of TIL therapy and ultimately CNET therapy. And this, of course, will lend itself to developing a robust potency assay. What have we generated to date is we've shown that we can create products that are polyclonal. They'll always target multiple cancer antigens and will always target multiple clonal neoantigens. We can generate good CD4 T helper cells and good CD8 responses in all of our products. And by using a dendritic cell-based expansion, we can reduce the amount of IL-2 in our process and create a fitter cell. So just to go a little bit deeper on this concept of a potency assay, we know the regulatory authorities have been remarkably consistent in this manner, that they want to see a demonstration that your product contains an active component of specific identity and potency. The specific identity for us would be CNET, and we can describe that for each patient because we have that antigenic information. And for each product, we can calculate the CNET dose as a fraction of the overall T cells. So we think we can get to a specific identity. Can you get to a specific potency? Of course, potency is the delivery of a specific event, a specific result, and for us, of course, that will be a reduction in tumor mass, and we believe that that will be correlated to the presence of CNETs. And together, we can build a clearer picture of mechanism and a clearer idea of potency and release of product. And we think that will be a very important development for the field. So looking at some of that data in action. So here we're looking at a patient that was dosed with about 16 million CNET. 65% of the product was reactive for the clonal neoantigens targeting that patient. And as you can see, post-dosing, looking at sort of at day 3, 7, 10, you see that the CNETs expanded and started to engraft in the patient, but they sort of fall away post-day 10, so they didn't persist. We think that those kinetics are going to be very important when understanding the mechanism of TIL therapies and CNET therapies. And this isn't something that you can do with a standard TIL therapy in this manner because you don't have that antigenic information. So what we believe is that an increasing of the CNET dose will give us improved persistence and ultimately efficacy. And this was, of course, seen in the earlier days of the CAR-T field, where there were also subclinical doses being manufactured. So how does that play into our studies? We've got a lung cancer study, Chiron, that's 40 patients, excluding EGFR driver mutations and never smokers. Still, that's the vast majority of non-small cell lung cancer patients. We're looking at CNET initially as a monotherapy and evaluating all the markers of safety, tolerability, activity that you would expect, but also bringing in some unique elements like a bespoke ctDNA assay driven by our ability to track and assess the T cells at a much greater level of precision. And we're open and recruiting in UK, EU, and the US. Our Thetis or melanoma study follows a very similar principle. We have 40 patients in a monotherapy arm and looking to recruit up to 20 patients in a checkpoint combination arm for patients refractory to checkpoint inhibitor therapy. So looking at how that schema plays out, the patient is enrolled for therapy. You can actually procure tumor ahead of them going on first-line therapy, so ahead of checkpoint, while the patient's on a checkpoint, or indeed afterwards. And actually, we've done all three of those and manufactured products successfully from those different points in time. Once the patient has had a scan demonstrating progressive disease, we will bring them into the therapeutic side of the study, precondition them with cyclophosphamide from darabine, and then infuse the CNETs, followed by low-dose IL-2. It's worth noting that the level of chemo conditioning we do is significantly less intense than standard regimes that you would see in TIL therapy, and much more akin to what you would see in a CAR-T. So looking at the first 6 patients, there was 3 in lung, 3 in melanoma. There was a median of 2.5 lines of prior therapy, and all patients had progressive disease at the time of enrollment into the therapeutic side of the study with lymphodepletion. When we started the work, looking at a number of other T-cell therapies, we hypothesized that the active CNET dose, not total T-cell dose, but the CNET dose, should be somewhere between 10 to 500 million CNET. When we looked at the performance of our first process, we saw a clustering really towards the lower end of that range at 15 million CNET, and you can see on the right-hand side how those doses clustered. So we know that we're at the sort of lower end of our prospectively targeted range. What did we see in those first 6 patients? We were pleased with the tolerability. The Independent Data Safety Monitoring Committee has given us the green light to continue both studies, and so they are active and ongoing right now. And in terms of activity, we saw a stabilization of that progressive disease in 4 of the 6 patients. We saw tumor reduction in 1 patient that had the highest dose in 2 of 4 lesions, 2 of 4 splenic and hepatic lesion, reduction of 55 and 90%, which was sort of in the right direction and encouraging. And we saw evidence of engraftment, that sort of multiple time points of T cells expanding, CNET expanding in number in 3 of 6 patients. And we felt very encouraged that the platform of analytical tools we've built around our CNETs are robust and can track these cells in the patients. What was clear is that we do want to look at higher doses of the process, and that's what we set out to do with our process 2. So there's, broadly speaking, 2 steps in the process: pre-expansion and expansion. So that's T-cell harvest and dendritic cell-driven expansion. And we optimized our process 1 to improve the CNET dose to approximately 10x, 10 times the amount, by additional cytokine and protocol adjustments across these 2 key areas in the process, pre-expansion and expansion. And we haven't changed the end-to-end manufacturing time by doing that. Today, we're manufacturing out of the Royal Free Hospital, a facility that can generate about 50 doses. We'll bring our second facility online with regulatory approval. We're expecting the Q4 of this year, and that's at Cell Therapy Catapult, generating 200 doses a year. And our third facility, a large modular facility that would have capacity for in excess of 1,000 doses, that could run multiple different processes and multiple different indications at the Hayes facility coming online in 2023. Central to what we're doing is developing a platform, and we've talked a lot about the tumor product today, but we believe it's feasible to develop a product, a CNET product from blood. Also think there could be significant clinical utility in a gene-edited product, knocking down genes that might be beneficial for anti-tumor effect. And of course, we believe that T cells actually can be stored quite readily for prolonged periods of time, which will allow you to bank tumor and or other constituents of the products for prolonged periods of time in anticipation of the patient potentially needing them downstream. So in 21, we've generated 6 patients of data. We've opened clinical sites in the US and Europe and enrolled our first US patient. Plan to dose our first patient with a checkpoint combination. We'll be bringing that second GMP facility, the Cell Therapy Catapult, online, filing an IND for our third indication, which we expect to be head and neck squamous cell, and bringing out 10 patients of monotherapy data, and of course bringing some of that to SITC later this year. In 22, we'll dose and start dosing patients in our higher dose process 2 with a 10x improvement, looking at Q3 to bring the first sort of 5 patients of data in that process, and towards the end of the year, 10 patients of data with supporting translational science. We'll also be bringing around the middle of the year about 6 patients worth of data out with the checkpoint combination, and we'll be able to look at the contribution of both dose and the contribution of the checkpoint inhibitor to the CNET therapy efficacy, activity, persistence, and the features that we can track in the patient. So in closing, we have 2 ongoing clinical trials with near-term data readouts and the plan to bring new indications. We are targeting a novel class of antigen, the clonal neoantigen, and we can do so with exclusive access to TracerX. Our platform can deliver products that can target multiple cancer antigens present on all tumor cells, and we think the platform lends very well to developing a potency assay for the release of product. We have a robust and manufacturing system, we think readily applicable to the commercial setting, and cash to complete our planned clinical studies, expand the manufacturing capacity, and broaden the pipeline. Thanks for your attention.